Hello everyone, I am Prajna Jain, lecturer in microbiology. I have completed my MSc in medical laboratory technology in microbiology and immunology. So I'll be teaching you all the important topics coming under microbiology so that you will find it easier to understand the topics during your examination studies. First, we will start with virology. So I'll be covering entire virology in series. Today, we will start with basics of virology, which includes general properties of viruses, morphology of viruses, etc. So, what are viruses? These viruses are smallest unicellular microorganisms. So, there are various types of microorganisms, right? Bacteria, virus, fungus, parasite. Compared to all these microorganisms, viruses are the smallest ones, measuring about 20 to 400 nanometer in size. So, bacteria measure in micrometers, whereas viruses measures in nanometers. So approximately viruses are 1000 times smaller than that of the bacteria. And these viruses are obligate intracellular. Obligate means strict, and intracellular means they are living inside the cells of the host. Host may be human beings or animals. So these viruses are strictly living invading the cells and multiplying inside the cells of the host, and they are the most primitive microorganisms infecting the humans, and they differ from bacteria in many ways. There are various differences between bacteria and viruses. So, coming to general properties of viruses. This can be a 5 marks question. Usually this will be asked for 5 marks. List 5 general properties of viruses. So first property that we have already discussed is they are obligate intracellular. They strictly invade the cells of the host, and they possess either DNA or RNA but never both. So these viruses possess either DNA or RNA as the genetic material. They do not possess both. So depending upon the type of nucleic acid or genetic material present in the virus, they can be classified into DNA viruses and RNA viruses. There are various DNA viruses, variety of RNA viruses, but both DNA and RNA will not be there in the same virus. And third property is they are filterable. So as I have already told, bacteria are 1000 times bigger than that of the viruses, measuring in micrometers. So if we are using the bacterial filters, of course the viruses can be filterable because they are very small compared to the bacteria. Viruses can be filtered in the bacterial filters. And the fourth point is they cannot be grown on artificial cell-free culture media, but they can be grown in experimental animals, embryonated egg or tissue culture. Since viruses requires living cells for its growth and multiplication, they cannot be grown in the artificial culture media which are not containing the living cells. But rather than that, we can cultivate these viruses, we can grow these viruses in experimental animals or embryonated egg or by tissue culture. About all these viral cultivation process we can study in the upcoming classes. Then fifth point is viruses do not multiply by binary fission similar to bacteria, but they follow a complex method of multiplication. And the sixth point is they do not have any proper cellular organization. They don't have cell membrane, including ribosomes, mitochondria, endoplasmic reticulum. They don't even have enzymes which are necessary for protein and nucleic acid synthesis. They depend upon the host for all these, and they are not susceptible to antibacterial antibiotics. So antibiotics are basically the drugs which are used to treat the bacterial infections. So we cannot use antibiotics to kill the viruses. So to kill the viruses, we have to use antiviral drugs or interferons. So next, coming to structure or morphology of the general morphology of all the viruses. So viruses do not contain any cell organelles or enzymes necessary for its protein and nucleic acid synthesis, but viruses usually contain three parts. They are containing three components. That is, first one is innermost core, that is nucleic acid, either it may be DNA or RNA, and next to nucleic acid usually there will be capsid, and covering the capsid there will be envelope. And coming to nucleic acid, viruses have only one type of nucleic acid, that is either DNA or RNA. Based on the type of nucleic acid, and viruses are classified into DNA viruses and RNA viruses. So there are various DNA viruses like hepatitis B virus, adenovirus, variola virus, parvovirus B19, herpes virus, which includes herpes simplex virus, Epstein-Barr virus, cytomegalovirus, etc. So examples for RNA virus, which contains RNA as the genetic material, ribonucleic acid. So examples includes poliovirus, Coxsackie virus, hepatitis A virus, dengue virus, rubella virus, rabies virus, measles virus, mumps virus, influenza virus. There are various examples for RNA virus. So this is also one of the 3 marks questions. Give 3 examples for DNA virus or give 3 examples for RNA virus. And this nucleic acid may be single stranded or double stranded. It can be circular or linear. It may be segmented or unsegmented. So innermost core of the virus contains this genetic material, that is either DNA or RNA. Next to nucleic acid there will be capsid. So capsid, it is composed of number of repeated protein subunits called capsomers. So as you can see in the picture, this is a general structure of a virus. So innermost core contains nucleic acid. So here you can see single stranded nucleic acid, and next to nucleic acid there is a capsid which is composed of number of protein subunits called capsomers. So capsid is composed of capsomers. And what are these functions of the capsid? So mainly it protects this nucleic acid from the external environment. It mainly plays important role in protection of the nucleic acid, and it is antigenic and it is specific for each virus. Different virus contains different types of capsid, or it is specific for specific virus. And if the virus, so the third part, that is envelope, that is optional. Some of the viruses contains envelope, some of the viruses are non-enveloped. If the viruses are non-enveloped, then capsid plays important role in initiation of first step of multiplication. So it helps in attachment of the virus to the specific receptor on the host cell, which facilitates the entry of the virus. So virus has to invade the host cell, right? If the virus is lacking the envelope, if the virus is non-enveloped, then capsid plays important role in attachment to the host cell receptors. Thus, it helps in invading the host cell. Then, depending upon the arrangement of capsomers surrounding the nucleic acid, there are 3 types of symmetries of nucleocapsid, that is icosahedral symmetry, helical symmetry, and complex symmetry. And the outermost layer of the viruses, outermost component of the viruses is envelope. Some of the viruses are enveloped, some of the viruses are lacking this envelope, so they are non-enveloped. So basically this envelope is lipoprotein in nature. It contains lipid part and protein part. So this lipid part of this envelope, it is derived from the host cell membrane. After invading the host cell, it acquires the lipid part from the host cell membrane, and the protein part is virus coded, which is made up of subunits called peplomers or glycoproteins. So these peplomers are projected as spikes on the surface of envelope. As you can see in the picture, you can see the glycoprotein spikes surrounding the envelope. And based on the presence and absence of envelope, there are examples for enveloped viruses as well as non-enveloped viruses. So in this picture, you can see various types of symmetries of nucleocapsid. So there are three types of symmetries, right? One is icosahedral insymmetry. In this picture you can see the icosahedron shape of the nucleocapsid. And the next one is helical symmetry. Here the nucleic acid and capsid is helically coiled. And the third type of nucleic acid is complex symmetry, which is not, it doesn't have a proper type of symmetry, it is complex. So next, coming to size of the viruses. So we all know that viruses are extremely small. They measure about 20 nanometer to 400 nanometer in size, and the smallest known virus that is clinically important, it is parvovirus, which measures about 20 nanometer in size, and the largest virus is pox virus, which is 400 nanometer in size. So the pox virus is it is the first virus that is considered to be eradicated from the world, and the viruses can pass through the bacterial filters. So since the viruses measures in nanometers, we cannot visualize these viruses under light microscope or compound microscope. As we observe the bacteria under light microscope, we cannot observe the viruses under light microscope since they are extremely small. So how to observe these viruses under microscope? So we can use electron microscope to visualize the viruses, or ultracentrifugation, or we can use the membrane filters of different pore size. According to the size of the viruses, we can isolate the different viruses. And next, it is shape of the viruses. So most of the viruses are spherical or round in shape, but there are certain exceptions, like rabies virus is bullet shaped, and pox virus is brick shaped, and rotavirus is cartwheel shaped structure, and adenovirus appears like space vehicle shaped, and Ebola virus, it has filamentous shape. So you can all see the different types of structures of different viruses. So all these shapes we can make out under electron microscopy or immune electron microscope. So that's it for today. So just quickly I'll give the revision of today's topic. First, we had studied about what is virus. So it is a smallest unicellular microorganism measuring about 20 to 400 nanometer in size, and the general properties involves they are obligate intracellular, they possess either DNA or RNA but never both as the genetic material, and they can be easily filtered through bacterial filters because they are extremely small, and there should be a experimental animal or embryonated egg or tissue culture to cultivate the viruses. We cannot grow the viruses on artificial cell-free culture media like bacteria. So next it is viruses follow a complex method for replication. They do not follow binary fission like bacteria. Then they do not contain proper cellular organization. They don't have cell wall, cell membrane, mitochondria, ribosomes, endoplasmic reticulum, etc. They don't even have enzymes which are necessary for protein and nucleic acid synthesis. We cannot use antibiotics for treating the viral infection. And about structure, it basically contains nucleic acid, capsid and envelope. So nucleic acid, it may be of DNA or RNA, and the next to nucleic acid there will be capsid, which is composed of capsomers, and if the viruses are enveloped, they will be having envelope or at the outermost layer. If they are non-enveloped, this nucleic acid plays important role in attaching the virus to the receptors of the host cell, and this capsid has different. Nucleocapsid has different symmetries: icosahedral symmetry, helical symmetry, and complex symmetry. And envelope, it is surrounding the nucleocapsid, which is lipoprotein in nature, and it also has peplomers which project as spikes on the surface of envelope. And next, coming to size, they are extremely small, measuring about 20 to 400 nanometer in size. We have to use electron microscopy to visualize the viruses. And the shape, usually all viruses will be round or spherical in shape. There are some exceptions like rabies virus, which has bullet shape, pox virus has brick shape, rotavirus has wheel shape, adenovirus has space vehicle shape, and Ebola virus has filamentous shape. And that's it for today. So next we will be studying about more information on viruses. So thank you for watching. If you are having any queries, you can ask in the comment section.